So today we'll be discussing on our second chapter on the book of Cellular and Molecular Immunology. Chapter 2 will be Cell and Tissue of Immune System, whereby we'll discuss the cells of immune system. This include the phagocyte, mast cell, basophil, eosinophil, dendritic cell, and lymphocyte. But also we'll discuss the nature of killer cells, cytokine secreting inert lymphoid cells, the anatomy and function of lymphoid tissues that are bone marrow, thymus, lymphatic system, lymph node, spleen, cutaneous and mucous immune system. So let us start. The cell of innate and adaptive immune system are normally present as circulating cell in the blood and lymph and extravascular cells in lymphoid organs and scattered virtually in all tissues. The anatomic arrangement of these cells in lymphoid tissue and their ability to circulate and exchange among blood, lymph, and tissue are of critical importance for immune responses. The immune system faces numerous challenges to generate effective protective response against infectious agents. First, the immune system must be able to respond rapidly to small number of many different microbes that may be introduced at any site in the body. Second, in the adaptive immune response, there are very few naive lymphocytes that can specifically recognize and respond to any one antigen. Third, the effector mechanism of adaptive immune system, antibodies and effector T cell may have to locate and destroy microbes at sites that are distant from the site where the immune response was induced. The capacity of the immune system to meet these challenges and to optimally perform its protective function is dependent on remarkably rapid and varied response of immune cells, the way these cells are organized in lymphoid tissue, and the ability to migrate from one tissue to another. This chapter describes the cells and tissue that make up the immune system. In chapter 3, we describe the traffic pattern of lymphocytes throughout the body and the mechanism of migration of lymphocytes and other leukocytes. Cells of the immune system. The cell that serves specialized role in the innate and adaptive immune response are phagocytes, dendritic cells, antigen-specific lymphocytes, and various other leukocytes that function to eliminate antigen. These cells were introduced briefly in chapter 1. These cells are almost all derived from hematopoietic stem cell in the bone marrow, which differentiate along branching lineage based on their common precursor. Immune cells are broadly classified as either myeloid cell, which include phagocyte and most dendritic cell, or lymphoid cell, which include all lymphocytes. The number of some of these cell types in the blood are listed in table 2.1. Although most of these cells are found in the blood, the response of lymphocyte to antigen usually occur in lymphoid and other tissue, and therefore may not be reflected by changes in the number of blood lymphocytes. The expression of various membrane protein is used to distinguish distinct population of cells in the immune system. For instance, most helper T cells expresses a surface protein called CD4, and most cytotoxic T lymphocyte, CTLS, expresses a different surface protein called CD8. These and many other surface proteins are often called markers because they identify and discriminate between or mark different cell population. The function of the most of these markers in the cell on which they are expressed is now understood. The most common way to determine if a particular marker is expressed on a cell is to test if antibody specific for the markers bind to the cells. In this context, the antibody are used by investigators or clinicians as analytical tools. Hundreds of different pure antibody preparations called monoclonal antibodies are available, each specific for a different molecules and labeled with chemicals that can be readily detected on cell surface by use of appropriate instrument. Monoclonal antibodies are described in chapter 5, and method to detect labeled antibodies bound to the cell are discussed in appendix 3. The class of differentiation CD nomenclature is widely adopted uniform method for naming cell surface molecules that are sometimes characteristics of particular cell lineage or differentiation stage and are recognized by a group class of monoclonal antibodies. Thus, all antigenically distinguishable cell surface protein and some carbohydrate are given a CD number designation, e.g. CD1 or CD2. Although originally devised to define circulating immune cells, leukocyte, subtype CD4 markers are found on all cell types in the body. Cell surface molecules, now identified by CD number, have important role in immune responses and are the target of many therapeutic antibodies used in treatment of inflammatory disease and cancer. The Appendix 1 provides a current list of leukocyte CD marker that mentioned in this book. The white blood cell, we have normal blood cell count, mean number, and normal range. These are the ones that you can easily find in the book. We have the neutrophil, we have the white blood cell, leukocyte in general, we have eosinophil, we have basophil, we have lymphocytes. They are listed in range that you can see yourself in the book. The phagocytes. Phagocytes, including neutrophils and macrophage, are cells whose primary function is to ingest and destroy microbes and remove damaged tissue. The functional response of phagocyte in host defense consists of sequential steps that recruiting of the cell to the site of infection, recognition of and activation of microbes, engagement of the microbes by a process of phagocytosis, and destruction of ingested microbes. In addition, through direct contact and by secreting cytokines, phagocytes communicate with other cells in the way that promote or regulate immune responses. Blood neutrophil and monocyte, which differentiate into macrophage after entering tissue, are produced in the bone marrow, circulate in the blood, and are recruited in site of inflammation. Although both are actively phagocytic, they differ in significant ways. You will see table 2.2. The neutrophil response is more rapid, and the lifespan of these cells after the interstitial is short, whereas the macrophage in tissue can live for long periods so that the macrophage response may last for prolonged time. Neutrophils mainly use cytoskeletal rearrangement and enzyme activation to mount rapid transient response, whereas macrophage response rely more on introduced gene transcription and protein expression. In addition, as we discuss later, there are subset of macrophage that normally reside in healthy tissue, but neutrophils do not. The function of phagocytes are important in innate immunity, we'll see in chapter 4, and also in the effector phase of some adaptive immune response, see chapter 10. As prelude to more detailed discussion of the role of phagocyte in immune response in later chapter, here we will describe the development and morphological features of neutrophil and macrophage and briefly introduce their function response. Distinguished properties of neutrophil and macrophage. Neutrophil and macrophage. Originally, these are the neutrophil are coming from hematopoietic bone marrow. Macrophage, they are blood monocyte derived from bone marrow in inflammatory reaction. They are many tissue-resident macrophage derived from stem cell in yolk sac or fetal liver early in development. Lifespan for neutrophil is 1 to 2 days. Macrophage, inflammatory macrophage, days or weeks. It can stay days to weeks. Response to activating stimuli. For neutrophil, it's rapid, short-lived activation of preformed enzymes. For macrophage, more prolonged, slower, often dependent on the new gene transcription. Phagocytosis. For neutrophils, rapid ingestion of microbes. For macrophage, prolonged ability to ingest microbes, apoptotic cell, tissue debris, and foreign material. Reactive oxygen species. For neutrophil, rapidly induced by assembly of phagocyte oxidase or respiratory burst. For macrophage, less prominent. Nitric oxide for neutrophil are low level or none. For macrophage, induced after transcription or activation of ionos. Degranulation. Neutrophil major response induced by cytoskeletal rearrangement. For macrophage, degranulation is not prominent. Cytokine production. Neutrophil, low level per cell. For macrophage, major function activities, large amount per cell require transcription activation of cytokine gene. Knot formation, rapidly induced by extraction of nuclear content, knot. Pyrostosis, no pyrostosis in neutrophil, but prominent, caspase-1 activation. This table has listed the major difference between neutrophil and macrophage. The reactions summarized above are described in the text. Note that the two cells share many features such as phagocytosis, chemotaxis, and ability to migrate through blood vessels into tissue. HSC is hematopoietic stem cell. Ionos is inducible nitric oxide synthetase. NET is neutrophil extravascular trap. Now let us discuss neutrophil. Neutrophil are the most abundant population of circulating white blood cell and the principal cells type in acute inflammatory reaction. Neutrophils circulate as spherical cells approximately 12 to 15 micrometer in diameter with numerous membrane projections. The nucleus is segmented into 3 to 5 connected globules. Because of the nuclear morphology, neutrophils are called polymorphonuclear lymphocyte, PMNs. To contrast them with the mononuclear cell, e.g. macrophage and lymphocyte, whose nuclei are not lobed. The cytoplasm contains two types of membrane-bound granule. The majority of these granules, called specific granules, are filled with enzymes such as lysosomes, collagenase, and elastase. These granules do not stain strongly with either basic or acidic dye, hematoxylin and eosin respectively, which distinguish neutrophil from two other types of circulating leukocyte with cytoplasm granules called basophil and eosinophil. The remainder of granules of neutrophil called azurophilic granules because they are stained by azu A dyes, contain enzyme e.g. myroperoxidase and microcidal substances including defensins and cathelicidin, which we will discuss in chapter 4. Neutrophils are produced in bone marrow and arise from precursor that also give rise to circulating monocyte. Production of neutrophil is stimulated by granulocyte, colony stimulating factor, and granulocyte macrophage colony stimulating factor. An adult human produces more than 1 times 10 power 11 neutrophil per day, each of which circulate in the blood for a few hours or up to 5 days before dying. Neutrophil may migrate to site of infection rapidly after entry of microbes. After entering tissue, neutrophil function for only 1 to 2 days, and most of them die. The major function of neutrophil is to phagocytize microbes, especially opsonizing microbes and the product of necrotic cells, and destroy these in phagocytosomes. In addition, neutrophil may secrete granules content and also extrude their nuclear content, forming neutrophil extravascular trap, which is served to immobilize and kill extracellular microbes, but also many damage health tissue. Mononuclear phagocyte. The mononuclear phagocyte system includes circulating bone marrow derived cells called monocyte, many of which become macrophage when they migrate into tissue. The tissue-resident macrophage, which are initially derived from yolk sac or hematopoietic precursor during fetal life. Development of macrophage and monocyte. After birth, the cell of monocyte macrophage lineage arises from committed precursor cell in the bone marrow, driven by cytokine called monocyte or macrophage colony-stimulating factor. These precursors mature into monocyte, which enter and circulate in the blood, where they have a short lifespan of approximately 1 to 7 days. Blood monocytes are efficiently recruited into tissue site of infection or injury, and therefore most macrophages at site of inflammation are monocyte-derived. Most long-lived tissue-resident macrophages are derived not from the bone marrow, but from the yolk sac or fetal liver precursor during fetal development. These cells have self-renewal capacity, so they can maintain stable number. They often assume specialized phenotypes depending on the organs. Examples are Kupffer cell in lining of sinusoid in the liver, alveolar macrophage in the lung, and microglia cells in the brain. In the steady state, blood monocytes are recruited at low rate into healthy tissue, where they differentiate into tissue-resident macrophage. This pathway of monocyte differentiating into tissue macrophage supplement the self-renewal of the fetally derived cells and account for varying fractions of resident macrophagy in different tissue. Subset of monocyte. Monocytes are 10 to 15 micrometer in diameter. They have been shaped nuclear and finally granule cytoplasm containing lysosomes, phagolytic vacuoles, and cytoskeletal filament. All human monocyte expresses class 2 major histocompatibility complex molecule, CD11b, CD86, and all mouse monocyte expresses CD11, 115, CD11b, and CD64. However, monocytes are heterogeneously and consist of different subsets distinguishable by cell surface marker and function, but not by morphology. In both human and mice, the most numerous monocyte, called classical or inflammatory monocyte, comprises of 90% to 95% of the blood monocyte in the humans, produce inflammatory mediator, are phagocytic, and are rapidly recruited to the site of infection or tissue injury. The second type of circulating monocyte, called the non-classical monocyte, as 0 to 10% of the blood monocyte, are recruited into the tissue after infection or injury and may contribute to repair. Classical or inflammatory monocytes are most often distinguished from non-classical monocytes by their relatively high expression of CD14 human, Li6C, and CCR2 mice. Some non-classical monocytes are known to claw along endothelial surface, described as patrolling, where they scavenge luminal microparticle and may play a role in eliminating circulating microbes and in repairing endothelial barrier defect. The developmental relationship between monocyte subset is not fully understood. Function of macrophage. Macrophages play a crucial role in innate and adaptive immune responses to infection and in repair of damaged tissue. A major function of monocyte-derived macrophage in the host defense is to ingest microbes by the process of phagocytosis and then to kill the ingested microbes. The mechanism of phagocytosis and killing, which we will discuss chapter 4, include formation of cytoplasmic membrane bond organelles that contain the microbes and fusion of these organelle with lysosomes. The enzymatic generation of reactive oxygen and nitrogen species in the lysosome that are toxic to microbes, and the digestion of microbial protein by proteolytic enzymes. Tissue-resident macrophage function as sentinel cell that sense the presence of microbes and respond by secreting cytokines that initiate and then amplify the protective response against the microbes. Some of these cytokines act on endothelial cell lining blood vessels to enhance the recruitment of monocyte and other leukocytes from the blood into site of infection. Other cytokines made by activated macrophage act on leukocyte and stimulate their migration to tissue site of infection or damage. Some important macrophage-derived cytokines are discussed in Chapter 4. Macrophages that have engulfed microbes can be induced by microbial molecule to undergo an inflammatory form of death called the pyroptosis, which usually results from activation of cytoplasmic enzyme complex called the inflammasome, which is discussed in Chapter 4. Pyroptosis lead to the release of cytokines that enhance the host inflammatory response to infection. In addition to ingestion microbes, macrophages ingest necrotic host cell, including cells that die in tissue because of the effects of toxin, trauma, and interrupted blood supply, and neutrophils that die after accumulation at site of infection. This is part of the cleaning process after infection and or sterile tissue injury. Macrophage can specifically recognize and engulf cells that die by apoptosis before the dead test cell can release their content and induce inflammatory response. This clearance by macrophage of apoptotic cells, including apoptotic neutrophil, is the process called the efferocytosis. Throughout the body and throughout the life of an individual, unwanted cells die by apoptosis and part of many physiological processes such as development or renewal of healthy tissue and maintenance of cell number or tissue homeostasis, and the dead cells are eliminated by macrophage. Macrophage serves as antigen-presenting cell that display fragment of protein antigen to T lymphocyte and activate T cell recruited to site of injury or infection. This function is important in effector phase of T cell mediated immune response. Chapter 6 and Chapter 10. Macrophage promote the repair of damaged tissue by stimulating new blood vessel growth, the angiogenesis, and synthesis of collagen-rich extracellular matrix fibrosis. These functions are mediated by cytokines secreted by macrophage that act on various tissue cells. Monocyte-derived macrophage may respond to microbes nearly as rapidly as neutrophils do, but macrophages survive much longer at site of inflammation. Unlike neutrophil, macrophage can undergo cell division at an inflammatory site. Therefore, macrophages are the dominant effector cell of the later stages in innate immune response several days after an infection. The macrophage receptors and activation. Macrophages are activated to perform their function by recognizing many different kinds of microbial molecules, as well as host molecule produced in response to infection and injury. These various activating molecule bind to specific signaling receptor located on surface of macrophage. Examples of these receptors are toll-like receptor, which are important in innate immunity and will be discussed in detail Chapter 4. Macrophages are also activated when other plasma membrane receptors bind to opsin on the surface of microbes. Opsinin are substances that coat microbial cells or other particles and tag them for phagocytosis. Examples of opsin receptors are complement receptors, which bind fragments to complement protein attached to microbial surface, and immunoglobulin G, IgG, Fc receptor, which bind to one end of IgG antibody molecule that already have microbes born at other end, discussed in chapter 13. Macrophagy phagocytosis of host cell is prevented in part by an inhibitory receptor on the macrophage called SIRP alpha, which recognizes CD47, a membrane protein on health cells that function as "don't eat me" signal. When CD47 binds to SIRP alpha, inhibitory signals are generated in the macrophage that prevent phagocytosis. In adaptive immunity, macrophage antimicrobial function are activated by some T lymphocyte, cytokine, and membrane protein that bind to signaling receptors on macrophage membrane. Subsets of macrophage. Macrophage can acquire distinct function and capability depending on the types of activating stimuli to which they are exposed. The clearest example of this is the response of macrophage to different cytokines made by a subset of T cell. Some of these cytokines activate macrophage to become efficient at killing microbes, called classical activation, and these cells are called M1 macrophage. Other cytokines activate macrophage to promote tissue remodeling and repair, called alternative activation, and these cells are called the M2 macrophage. These different pathways of activation and cytokine involved are discussed in chapter 10. The relationship between blood monocyte subset, discussed earlier, and macrophage subset is not well understood. Macrophage may also assume different morphological form after activation by external stimuli such as microbes. Some develop abundant cytoplasm and are called epithelioid cell because of their resemblance to epithelial cell of the skin. Activated macrophage can fuse to form multinucleated giant cell, which occur frequently in certain types of microbial infections, such as with the Mycobacterium and in response to indigestible foreign bodies. Mast cell, basophil, and eosinophil. Mast cell, basophil, and eosinophil are three additional cell types that play roles in innate and adaptive immune response. All three share the common property of having cytoplasmic granules filled with the various inflammatory and antimicrobial mediators, which are released from the cell upon activation. Another common feature of this cell is their involvement in immune response that protect against ailment and reaction that cause allergic disease. We will introduce the feature of this cell in this section and discuss their function in more detail in chapter 20. Mast cell. Mast cells are bone marrow-derived cells that are most abundant in the skin and mucosal epithelium. Upon activation, they release many potent inflammatory mediators that defend against infection by helminthic parasites or cause symptoms of allergic disease. A cytokine called stem cell factor, c-KIT ligand, is essential for mast cell development. Normally, mature mast cells are not found in circulatory, but are present in tissue, usually adjacent to small blood vessels and nerves. The cytoplasm contains numerous membrane-bound granules, which are filled with preformed inflammatory mediators such as histamine and acidic proteoglycan that bind basic dyes, impacting a dark blue color to the granules when special stains are used. Various stimuli can activate mast cell to release a cytoplasmic granules content into extravascular space, as well as to synthesize and release cytokine and inflammatory lipid mediator. The released histamine and other mediator promote changes in blood vessels that cause inflammation. Mast cell expresses high affinity plasma membrane receptor for a type of antibody called IgE and are usually coated with these antibodies. When the antibodies on the mast cell surface bind antigen, Signaling events are induced that lead to the mast cell activation. Mast cells are also activated when they recognize microbial product independent of IgE, and in this way, they function as tissue sentinel of innate immune system. Basophil. Basophil are blood granules with many structural and functional similarities to the mast cell. Like other granules, basophil are derived from hematopoietic precursor, mature in bone marrow, from progenitor distinct from those of the mast cell, and circulate in the blood. Basophil constitute less than 1% of the blood leukocyte. Although they are normally not present in tissue, basophil may be recruited to some inflammatory site. These cells are also contain granules that bind basic dye, and they are capable of synthesizing many of the same mediators as mast cell. Like the mast cell, basophil expresses the IgE receptor, bind IgE, and can be triggered by antigen binding to IgE. Because basophil numbers are low in tissue, their importance in host defense and allergic reaction is uncertain. Eosinophil. Eosinophil are granulocytes that expresses cytoplasmic granules containing enzymes that are harmful to the cell wall of parasites but also can damage host tissue. Eosinophil granules contain mainly basic protein that bind acidic dyes such as eosin and that appear red in stained blood smear and tissue section. Eosinophils are bone marrow derived and circulate in the blood from where they may be recruited into tissue. The cytokine GM-CSF, interleukin-3, interleukin-5 promote eosinophil maturation from myeloid precursor. Various membrane receptor on eosinophil, including Fc receptor for IgA, IgG, toll-like receptor, and interleukin-5 receptor, can generate signal that activate cell to raise their granulose content. Some eosinophils are normally present in peripheral tissue, especially in the mucosal lining of the respiratory, gastrointestinal, genitourinary tract, and their number can increase by recruitment from blood in the setting of inflammation. Dendritic cell. Dendritic cells are tissue resident and circulating cells that detect the presence of microbial and initiate innate immune defense reaction, and they capture microbial protein for display to T cell to initiate adaptive immune response. These cells are named because of their long membranous projection, reminiscent of dendrites of neurons. Most dendritic cells are widely distributed in lymphoid tissue, mucosal epithelial, and organ parenchyma. The location of dendritic cells in epithelial and tissue where microbes enter, their ability to acquire antigen and take them to lymph node where naive T cells circulate, and their rapid response to microbes all place these cells in unique position in immune system, serving as sentinel of infection that begin the rapid innate response but also link the innate response with development of adaptive immune responses. We will discuss the role of dendritic cell as mediator of innate immunity and as antigen-presenting cell in chapter 4, 6 respectively. Here we will introduce the general properties of dendritic cell. Development and features of dendritic cell subset. Subsets of dendritic cell can be defined on the basis of different cell surface markers, transcriptional factors, development from different precursor cells, tissue localization, and function. We will describe the major subsets that are important in immune responses and are distinguished from one another by their function and development and by the expression of different surface molecules and transcription factors. The common properties of these dendritic subsets include developmental dependence on cytokine, FLT3L, expression of CD11C protein, and the ability to present antigen to and activate naive T cell or induce T cell tolerance. Classical dendritic cell, also called the conventional dendritic cell, CDCS, are the major types of dendritic cell involved in capturing protein antigen of microbes that enter through epithelial barrier and presenting them to T cell. Classical dendritic cells were first identified by their morphology and ability to stimulate strong T cell response and are the most numerous dendritic cell subset in epithelial and lymphoid organ. They arise from bone marrow, hematopoietic stem cell, through development pathway that induce common precursor of both monocyte and classical dendritic cell, some of which develop into committed precursor for a classical dendritic cell called pre-classical dendritic cell. All these steps take place in the bone marrow. The pre-classical dendritic cell migrate to the peripheral tissue where they mature into a classical dendritic cell. Similar to tissue macrophage, these dendritic cells constantly sample the environment in which they reside. Classical dendritic cell may be further divided into two main subsets called major or classical dendritic cell and cross-presenting classical dendritic cell. Classical dendritic cell is the most numerous subset and is potent at capturing exogenous antigen, including CD4 T cell response. The classical dendritic cell subset is specialized to present antigen to naive CD8 T cell by a process called cross-presentation, discussed in Chapter 6. This subset can also present antigen to CD4 cell. Plasmacytoid dendritic cell, P dendritic cell, produce antiviral cytokine type of interferon in response to viruses and may capture blood-borne microbes and carry their antigen to spleen for presentation to T cell. These dendritic cells are named because after activation, they begin to resemble plasma cells morphologically. They develop in the bone marrow from the precursor distinct from that for classical dendritic cell and are found in the blood and in small number in lymphoid organ. Plasmacytoid dendritic cells are the body's major producers of cytokine called type 1 interferons, which have potent antiviral activities and play an important role in innate host defense against viruses. Monocyte-derived dendritic cell, more dendritic cell, include cells with functions similar to those of dendritic cell, but are derived from monocytes that were recruited into tissue inflammatory site. They express CD11C like all dendritic cell, and also monocyte markers such as CD11B and CCR2. Langerhans cells are dendritic cells found in epidermis that share function with classical dendritic cell, but are developmental related to tissue-resident macrophage, arising from embryonic fetal liver and yolk sac precursor. They are identified by their location and morphology in the skin, the presence of tennis racket-shaped cytoplasm organelle called Barbeck granules that are visible using electron microscope, and the expression of various markers. Langerhans cells may function in the context of skin infection to present antigen to and activate CD4, or in absence of infection to present self-antigen to CD4 cells and induce tolerance to these antigen. Another population of cells called the follicular dendritic cell have dendritic morphology but are not derived from bone marrow precursor, do not present protein antigen to T cell, and should not be confused with dendritic cell. Follicular dendritic cells are involved in B cell activation in the germinal center of secondary lymph node organs. See chapter 12. Lymphocyte. Lymphocytes, the unique cell of adaptive immunity, are the only cell in the body that expresses clonally distributed antigen receptors, each specific for a different antigenic determinants. Each clone of T and B lymphocyte expresses antigen receptors with a single specificity, which is different from specificities of receptor in all other clones. As we shall discuss here and later chapters, there are millions of lymphocyte clones in the body, enabling any individual to recognize and respond to millions of foreign antigens. The role of lymphocyte in mediating adaptive immunity was established by several lines of evidence accumulated over decades of research. One of the earliest clues came from the observation that humans with congenital and acquired immunodeficient state had reduced the number of lymphocytes in their peripheral circulation and in lymphoid tissue. Experiments done in mice and rats showed that depletion of lymphocytes impaired the response to immunization, and lymphocytes are the only cells type that can transfer specific immunity to microbe from immunized to naive animal. In vitro experiments established that stimulation of lymphocyte with antigen lead to the response that show many of the characteristics of immune responses induced under more physiological conditions in vivo. After identification of lymphocyte as mediator of humoral and cellular immunity, many discoveries were made at a rapid pace about different types of lymphocyte, their origin in bone marrow and thymuses, their role in various immune responses, and the consequences of their deficiency. Among the most important finding was that clonally distributed, highly diverse and specific receptor for antigen are produced by lymphocyte but not by any other types of cells. More recently, an enormous amount of information has accumulated about lymphocyte genes, proteins, and function. One of the most fascinating aspects of lymphocyte biology is how that the extremely diverse repertoires of antigen receptor with different specificity is generated from the limited number of genes for these receptors that are present in the germline. It is now known that the gene encoding the antigen receptor of lymphocyte are formed by recombination of DNA segments during the maturation of these cells. There is a random aspect of this somatic recombination event that results in the generation of millions of different recombined receptor genes and a highly diverse repertoire of antigen specificity among different clones of lymphocytes. The total number of lymphocytes in healthy adults is approximately 5 times 10 power 11. Of these, approximately 2% are in the blood, 4 in the skin, 10 in the bone marrow, 15 in the mucosal lymphoid tissue of the gastrointestinal and respiratory tract, and 65 in lymph node or lymphoid organ, mainly the spleen and lymph nodes. We first describe the properties of these cells and then their organization in various lymph nodes. So, classical conversion of dendritic cell. This was human dendritic cell, just a description if you want to follow it, the structure and how it looks like. There are different tables that have been expressed. Just consult the book to see it. Classes of lymphocyte. Lymphocyte consists of distinct classes with different function and protein products. The major classes of lymphocyte were introduced in chapter 1. Morphologically, all lymphocytes are similar, and their appearance does not reflect the heterogeneity or their diverse function. B lymphocyte, the cell that produce antibody were so called because in bed they were found to mature in organ called the buster of Fabricius. In mammal, no anatomic equivalent of Bartha exists, and the early stage of B cell maturation occurs in bone marrow. Thus, name B lymphocytes now refer to bone marrow-derived lymphocyte. T lymphocyte, the mediator of cellular immunity, arises from precursor cell in the bone marrow, which migrate to and mature to thymus. T lymphocytes refer to thymus-derived lymphocyte. They are a subset of B and T lymphocyte with distinct phenotypic and functional characteristics. Subset of B lymphocyte. The major subset of B lymphocyte are follicular B lymphocyte, marginal zone B lymphocyte, and B1 cells, each of which is found in distinct anatomical location within lymphoid tissue. Follicular B cells, the most numerous type of B cell in the body, are found in lymphoid tissue and blood. They express highly diverse, clonally distributed set of antibodies that serves as cell surface antigen receptor and as the key secreted effector molecule of adaptive humoral immunity. Follicular B cell give rise to the most highly affinity antibodies and memory B cell that protect people from repeat infection by the same microbes. In contrast, B1 and marginal zone B cells make up a minority of B cell and produce antibodies with limited diversity. B1 cells are found mainly in mucosa tissue and peritoneal or pleural cavity, where marginal zone B cells are present only in spleen in the rodent, but can be found in a circulation of human. Subset of T lymphocyte. The two major T cell subsets are defined by cell surface expression of CD4 and CD8 proteins. T cells are mediators of cellular immunity. CD4 T cells are helper T lymphocyte or their naive precursor, and CD8 T cells are cytotoxic lymphocytes or their precursor. Both CD4 and CD8 T cells expresses antigen receptor called alpha-beta T cell receptor. CD4 T cells helper T cells secrete cytokines that act on various other cells, including other T lymphocytes, B cell, macrophage, B cell and macrophage. CD8 cytotoxic lymphocyte recognize and kill infected cell infected with viruses and microbes that can live inside host and also kill cancer cell. CD4 regulatory T cell are third subset of cell expressing alpha-beta receptor. Their function is to inhibit immune response. In addition, natural killer cell, mucosa-associated invariant T, mite cell, and alpha-delta T cell are three numerous small subset of T cell that expresses T cell receptors with limited diversity analogous to the antibody made by B1 cell, and function of these classes of B and T cell will be discussed in later chapters. So here we have lymphocyte classes, like we have B lymphocyte, whereby a follicular B cell, which is responsible for antibody production, humoral immunity, the antigen receptor and specificity, the surface IG diversity specificity for many types of molecules. The selected phenotype marker for these follicular B cells are class 2 MHC, CD19, and CD23. When you look for marginal zone B cells, they are responsible for antibody production. Their surface IG and selected markers are IgM and CD27. When we talk of B1 cell, they are responsible for antibody production. The surface marker, they are CD43, 20, 27, but CD70 negative. When we speak of T lymphocyte, CD4 helper T lymphocyte is responsible for B cell activation, humoral immunity, macrophage activation, and cell-mediated immune stimulation. The selected phenotype are alpha-beta heterodimer, diverse specificity. They have marker of CD3, CD4, and CD8 negative. Talking of CD4, they are responsible for killing of cell infected with intracellular microbes. They are heterodimer, diverse specificity for peptide class 1. The marker they express are CD3, CD4 negative, and CD8 positive. Regulatory T cell, they suppress the function of other cells and other immune cells, regulation of immune response maintenance, self-tolerance. They are alpha-beta heterodimers, specifically for self and some for foreign antigen, peptide class 2. They express CD3, CD4 plus CD25, FOX, P3, most common, but other phenotype as well. The natural killer cells, they act as suppressive on or activate innate and adaptive immune response. Heterodimer limited specificity with alpha beta, and the markers for natural killer cell are CD56, CD16, Fc receptor for IgG, and CD3, 56, 16. Alpha delta T lymphocyte, these are helper and cytotoxic function, the innate immunity. They have alpha delta heterodimer limited specificity for peptide and the non-peptide antigen. They express CD3, CD4, and CD8 variable. They are rare. So that was just a summary of lymphocytes in table that you can find in your book and read it carefully. There are some other aspects that I've not read because I found it you cannot get a big picture, but I read what is important, that you can get picture or you can go together to have different formalities to understand this concept here. Development of lymphocytes. Lymphocyte, like all blood cell, arise after birth from the stem cell in the bone marrow. The origin of lymphocyte from bone marrow progenitors was first demonstrated by experiment with radiation-induced bone marrow chimeras. Lymphocytes and their precursor are radiosensitive and are killed by high dose of alpha irradiation. If a mouse of one inbred strain is irradiated and then injected with bone marrow cells or small number of hematopoietic stem cell of another strain that can be distinguished from the host, all lymphocytes that develop subsequently are derived from bone marrow cell or hematopoietic cell of the donor. Such approach have proved useful for examining maturation of lymphocyte and other blood cells. All lymphocytes go through complex maturation stages during which they express antigen receptor and acquire the functional and phenotypic characteristics of mature cell. The anatomical sites where the major steps in lymphocyte development occur are called generative or primary or central lymphoid organs. These include the bone marrow, where the precursor of all lymphocytes arise and B cell mature, and thymus, where T cell mature. We will discuss the process of B and T lymphocyte maturation in detail in chapter 8. Population of lymphocytes distinguished by history of antigen exposure. Naive lymphocytes that have matured in the bone marrow or thymus migrate to the secondary peripheral lymphoid organ, where they are activated by antigen to proliferate and differentiate into effector and memory cells. The mature T cells that emerge from the thymus are called naive T cells. B cells undergo most of their development in bone marrow, but the final step that generates mature naive B cell occur in spleen. Naive lymphocytes are functionally quiescent, but after activation by antigen, they proliferate and go through dramatic changes in phenotypic and functional activities. The activation of naive lymphocytes follow a series of sequential steps beginning with the synthesis of new proteins such as cytokine receptors and cytokines, which are required for many of their subsequent changes. The cell when will then undergo proliferation, resulting in an increasing size of the antigen-specific clone, a process called clonal expansion. In some infections, the number of microbe-specific T cells may increase more than 50,000 times fold within a week. And the number of specific B cells may increase up to 5,000 folds. This rapid clonal expansion of microbe-specific lymphocyte is needed to keep pace with the ability of microbes to rapidly replicate. Concurrently with proliferation, antigen-stimulating lymphocytes begin to differentiate into effector cells, whose function is to eliminate the antigen. Many of the effector cells migrate to the tissue site of infection, and some stay in secondary lymphoid organ. Other progenitors of antigen-stimulating B cell and T cell lymphocytes differentiate into long-lived memory cells, whose function is to mediate rapid and enhanced secondary response to subsequent exposure to antigen. Naive, effector, and memory lymphocytes can be distinguished by several function and phenotypic criteria seen in Table 2.5. The detail of lymphocyte activation and differentiation as well as function of each of these populations will be addressed later in the book. Here we will summarize the phenotypic characteristics of each population. The naive lymphocyte. Naive lymphocytes are mature T or B cells that have never encountered foreign antigen. The term naive refers to the idea that these cells are immunologically inexperienced. Naive lymphocytes are found in circulation and secondary lymphoid organs. Naive and memory lymphocytes are both called resting lymphocytes because they are not actively dividing or performing effector function and are in the state of rest or in the GO stage of the cell cycle before antigenic stimulation. Naive or memory B and T lymphocyte cannot be readily distinguished morphologically, and both are often called small lymphocyte when observed in blood smear. A small lymphocyte is 8 to 10 micrometer in diameter and has larger nuclei with dense heterochromatin and a thin rim of cytoplasm that contain few mitochondria, ribosomes, and lysosomes, but no visible specialized organelle. Naive or memory lymphocytes rely mainly on oxidative phosphorylation and fat acid oxidation to maintain their basal energy requirement. So characteristics of naive, effector, and memory lymphocyte. T cell. Naive, activated, and memory. T cell migration, they preferentially secondary lymphoid organs. So the effector are activated preferentially to inflamed tissue. Memory cells, they are preferentially to the inflamed tissue or mucosa tissue. Frequency of the cell response to particular antigen, naive, very low. Effector activated, very high. Memory, very low. Effector function, for naive, none. For activated or effector, cytokine secretion or cytotoxic activities. For memory, none. Cell recycling, no for naive, yes for activated cell, and yes or no for memory cell. Surface protein expression. So, for surface expression, we see interleukin 2R, CD5. They are low in naive, high in activated, and low in memory. L-selectin or CD62L is high in naive cell, low in activated effector cell, and variable in memory cell. For interleukin 7R or CD127, it is moderately high in naive cell, low in activated or effector cell, and high in memory. Adhesion molecule integrin or CD and CD44 is low in naive cell, high in activated effector cell, and high in memory cell. Chemokine receptor CCR7 is high in naive cell, low in activated effector cell, and variable in memory cell. Major CD45 isoform, human only, CD45RA is high in naive. CD45RO is high in activated. CD45RO is variable in memory cell. Morphologically, the naive cells are small and scant cytoplasm. Activated, they are large, more cytoplasm. Memory, they are small. Coming to B lymphocytes. These are IG producing. The membrane IgM or IgD. Frequently, when we talk of naive, the membrane for a B cell is IgM and IgD. On talk of activated or effector B cell, frequently secreted IgG, IgA, or IgE. In memory, we have IgG, IgA, or IgE. Talking of affinity of IgG, naive B cell is relatively low. Activated is increased during immune response, and memory is relatively high. Effector function, there are none for naive, and antibody secretion for activated or effector, and none for memory. Morphology is small and scant cytoplasm in naive, large more cytoplasm in activated, and small in memory cells. Surface protein expression. Chemokine receptor CXR5 is high in naive B cell and low in effector or activated B cell, and we don't know in memory cell. CD27 is low in naive cell, high in activated, and high in memory cells. So that was the summary of B cell and T cells, looking at their naive, activated effector cell or memory cell. We looked at differently in their characteristics to see what is happening to them. You can follow and read carefully in the book as you will be able to purchase it down. Naive lymphocytes typically live for 1 to 3 months. Their survival requires signal from antigen receptor and cytokine. The need for antigen receptor expression to maintain the pool of naive lymphocytes in secondary lymphoid organ was demonstrated in studies with mice in which the gene that encode the antigen receptor B cells or T cells were detected after lymphocyte had matured. In these studies, naive lymphocytes that host their antigen receptor died within 2 to 3 weeks. It has been shown that the antigen receptor of naive B cell generates survival signal even in the absence of antigen. Naive T lymphocytes recognize various self-antigen weakly enough to induce survival signal, but without triggering the stronger signal that are needed to initiate proliferation and into effector cell. Cytokines are also essential for survival of naive lymphocyte, and B and T lymphocyte expresses receptor for these cytokines. The most important of these cytokines are interleukin-7, which promote survival and low-level cycling of naive T cell and B cell activating factor. A cytokine belonging to tumor necrosis factor family, which is required for naive B cell survival. In the steady state or homeostasis, the pool of naive lymphocyte is maintained at a fairly constant number because the balance between spontaneous death of these cells and the production of new cell in the generative lymphoid organ. Any loss of lymphocyte lead to the compensatory proliferation of remaining ones and increased output from primary organ. This response of immune system to reestablish a normal total number of lymphocytes is called homeostasis proliferation. If naive cells are transferred into host that is deficient in lymphocyte, said to be lymphopenic, the transferred lymphocytes begin to proliferate and increase in number until they reach approximately the number of lymphocytes in the normal animal. Homeostatic proliferation appear to be driven by same signal that are required for maintenance of naive lymphocytes, namely the weak recognition of some self-antigen in the case of T cell or spontaneous B cell receptor signaling in B cell, and cytokines, mainly interleukin-7. This phenomenon is explored clinically in T cell therapy protocol as in treatment of some leukemias. The transferred T cell proliferate maximally if host T number are reduced, a process called lymphodepletion. See in chapter 18. Effector lymphocytes. In response to stimulation by antigen and other signals, naive lymphocytes enter the G1 stage of the cell cycle before going on to divide. Activated lymphocytes are larger, 10 to 12 micrometer in diameter, have more cytoplasm and organelles, and increase the amount of cytoplasmic RNA, and are called larger lymphocyte or lymphoblast. These changes require more energy and substrate for biosynthetic activities. The recently activated lymphocytes use aerobic glycolysis for energy and the tricarboxylic acid TCA cycle to generate the intermediate metabolic needed for new synthesis of protein, lipid, and nucleic acid. Some of these activated lymphocytes differentiate into effector lymphocytes that have ability to produce molecule capable of eliminating foreign antigen. Effector T lymphocytes include CD4 helper T cell and CD8 cytotoxic lymphocyte, and the effector B lymphocyte are antibody secreting cells, mainly plasmablast and plasma cell. Helper T cells activate B lymphocyte, macrophage, and dendritic cell by secreting cytokines that bind to receptor on these cells and surface molecules, such as CD40 ligands, CD154, which engage the CD4 on the cell. Cytotoxic lymphocytes have cytoplasmic granule filled with protein that, when released, kill the cell that the cytotoxic cell recognize, which are usually virus-infected cell or tumor cell. Both CD4 and CD8 effector T cell usually expresses surface protein indicative of recent activation, including CD25, a component of receptor for T cell growth factor interleukin-2, and altered pattern of molecule that mediate migration, selecting integrin chemokine receptor discussed in chapter 3. Majority of differentiated effector T lymphocytes migrate from secondary lymphoid organ where they were generated into tissue site of infection and short-lived. Many antibody-secreting B cells are morphologically identifiable in stained tissue section as plasma cell. They have characteristics nuclei placed eccentrically in cell with a chromatin distributed around the nuclear membrane in cartwheel pattern, abundant cytoplasm containing dense, rough endoplasmic reticulum that is in the site where antibody and other secretory and membrane protein are synthesized, and distinct perinuclear Golgi complexes where antibody molecules are post-translational modified to their final form and packed for secretion. It is estimated that half or more of messenger RNA in these cells code for antibody protein, and a single plasma cell can secrete thousands of antibody molecules per second. Plasma cells develop in lymphoid organ and at site of infection, and some of them migrate to bone marrows, mucosa tissue, where they may live and secrete antibody for long period after immune response is induced and even after antigen is eliminated. Plasmablasts are antibody-secreting cells with features of plasma cell, but they are capable of proliferation. They are found in circulation and can be identified by expression of CD19 and low level of typical B cell marker CD20 compared with the naive or memory B cells. Within a week after infection, a large number of plasmablasts can be detected in the blood, which secrete IgM, IgG, and IgA antibodies and are derived from the naive or memory B cell that were recently activated in secondary lymphoid organ. Some of these circulating plasmablasts are likely in transit from lymphoid organ to bone marrow and mucosal tissue, where they will remain as long-lived plasma cells. Memory lymphocyte. Memory cells are generated during infection but may survive in functionally quiescent or slowly cycling state for months or a year after microbes is eliminated. Some memory cells recirculate between blood and lymphoid tissue, similar to naive T cell, and others remain within non-lymphoid tissue without re-entering the blood for long period. Memory lymphocyte can be identified by their expression of surface protein that distinguish them from naive and recently activated lymphocytes, although it is still not clear which of these lymphocytes, this surface protein, are definitive marker of memory population. Memory T cell, like naive but not effector T cell, express high level of interleukin-7 receptor. Memory T cell also expresses molecules that regulate their migration into and out of lymphoid organ or tissue site of infection, and these vary depending to subset. See chapter 3. In human, most of naive T cell expresses a 200 kilodalton isoform of surface molecule called the CD45 that contain a segment of encoded by an exon designated A and is therefore called the CD4RA for restricted A. In contrast, most activated and memory T cell expresses 18 kilodalton isoform of CD45 in which the A axon RNA has been spliced out. This isoform is called CD45RO. However, this way, distinguishing naive from memory T cell is not perfect. The interconvention between CD45R plus and CDR plus population has been documented. The frequency of memory cell increases with age because individuals are continually exposed to foreign antigen, such as environmental microbes. Memory T cell make up less than 5% of peripheral blood T cell in newborn, but 50% or more in adult. As individual age, the gradual accumulation of memory cell compensate for reduced output of new naive T cell from thymus, which involute after puberty, are discussed later. Memory B cell lymphocyte may express certain classes isotope of membrane IgG, such as IgG, IgE, IgA, as a result of isotope switching, whereas naive B cell expresses only IgG, IgM, and IgD. See chapter 5 and chapter 12. In human, CD27 expression is the marker for memory B cell. Memory cells are heterogeneous and consist of subsets that differ, especially with respect to their location and migratory properties. Memory T and B cell will be discussed further in chapter 9 and chapter 12, respectively. The distinguishing feature of naive, effector, and memory lymphocyte reflect a different program of gene expression that are regulated by transcription factor and stable epigenetic changes, including histone methylation, acetylation, and chromatin remodeling. For example, a transcription factor called Kruppel-like factor 2, KL factor 2, is required for maintenance of naive T cell phenotype. The phenotype of functional different types of CD4 effector T cell called T helper 1, T helper 2, T helper 17 cell depend on a transcription factor TBET, GAT3, and RORI, respectively, as well as epigenetic changes in cytokine lock. Other transcription factors are required for maintaining the phenotype of memory B cell, B and T cell. Our understanding of molecular determinants of lymphocyte phenotype is still incomplete and evolving. Natural killer cell and cytokine secreting. Innate lymphoid cell. The innate immune system includes several developmentally related subset of bone marrow derived cell with lymphoid morphology and effector function similar to those of T cell but lacking T cell antigen receptor. The major function of these cells are to provide early defense against infections, pathogen, so to recognize stressed and damaged host cell and help eliminate these cells, and to influence the nature of subsequent adaptive immune response. Natural killer cells have cytotoxic activities similar to that of CD8 cytotoxic lymphocytes. They circulate in the blood and are present in various lymphoid tissue. Innate lymphoid cell produce cytokines similar to those secreted by CD4 helper T cell. The innate lymphoid cells can be grouped into three major subsets based on the cytokines they secrete, analogous to the three subsets of CD4 helper T cell described in chapter 10. Innate lymphoid cells are rare in the blood and are present mostly in tissue, especially mucosal tissue such as the lung and intestine. The common lymphoid progenitor in the bone marrow that give rise to the T and B lymphocyte also give rise to the precursor of both natural killer cell and innate lymphocyte cell. And natural killer cell and innate lymphocyte cell share expression of several lineage-specific markers and transcriptional factors. Lymphoid tissue-induced cells are the types of innate lymphoid cells that produce the cytokine lymphotoxin and tumor necrosis factor and are essential for formation of organized secondary lymphoid tissue, described later in chapter. Natural killer cell and innate lymphocyte cell are described more in detail in chapter 4. Anatomy and function of lymphoid tissue. The primary lymphoid organ, also called generative or central lymphoid organ, include the bone marrow and thymus, and are the sites where lymphocytes first express antigen receptor and attain phenotypic and functional maturity. B lymphocytes mature partially in the bone marrow, enter the circulation, migrate to the spleen where they complete their maturation, and then populate secondary lymphoid organs. T lymphocytes mature in the thymus and they enter the circulation and migrate to secondary lymphoid organ. Two important functions shared by generative organs are to provide growth factor and other molecular signal needed for lymphocyte maturation and to present self-antigen for selection of maturing lymphocytes. Secondary or peripheral lymphoid organs include the lymph nodes, spleen, and component of mucosa immune system are where lymphocyte response to foreign antigen are initiated and developed. These organs are anatomically organized in the way that optimize the cellular interaction necessary for initiation of adaptive immune responses. Lymphocyte and antigen-presenting cells are localized and concentrated in an anatomically defined area of this organ, which are also the site where foreign antigen are transported and concentrated. This ensures that antigen and antigen-specific naive lymphocyte can come together to initiate adaptive immune response. The anatomy of lymphoid organ also enables the T cell and B cell to interact after they are activated by antigen, as well as we'll discuss in chapter 3. Many lymphocytes constantly recirculate and exchange among the circulation, secondary organ and tissue. The bone marrow. Bone marrow is the site of generation of circulating blood cell, including the red blood cell, the granulocytes, monocyte, and site of B cell maturation. The generation of all blood cells called hematopoiesis occur initially during fetal development in blood island of the yolk sac and periocterial mesenchymal, then shift to the liver between the 3rd and 4th month of gestation, and finally shift to the bone marrow. At birth, hematopoietic takes place in bone marrow throughout the skeleton, but it becomes increasingly restricted in the marrow of the flat bone, so that by puberty, hematopoietic occur mostly in the sternum, vertebrae, iliac bone, and ribs. The red marrow that is found in these bones consists of sponge-like reticular framework located between long bone sides. Long bones consist of long bone trabeculae. The space in this framework contains a network of blood-filled sinusoid lined by endothelial cells attached to the discontinuous basement membrane. Outside the sinusoid are clusters of precursor of blood cells in various stages of development, as well as fat cells. The blood cell precursor mature and then migrate through the sinusoid basement membrane and between endothelial cells to enter the fascicular circulation. When the bone marrow is compromised or there is an exceptional demand for production of new blood cells, the liver and spleen often become site for extramedullary hematopoiesis. Red blood cell, granulocyte, monocyte, dendritic cell, mast cell, platelets, B and T cell lymphocytes, and ILC all originate from common hematopoietic stem cell in bone marrow. Hematopoietic stem cells are multipotent, meaning that a single hematopoietic cell can generate all different types of mature blood cells. Hematopoietic cells are also self-renewing because each time they divide, at least one daughter cell maintain a properties of stem cell and other can differentiate along particular lineage called asymmetric division. Hematopoietic stem cell can be identified by the presence of surface marker including the protein CD34 and c-KIT, and the absence of lineage-specific marker that expressed in the mature cell. Hematopoietic stem cells are maintained within specialized microscopic anatomic niche in the bone marrow. In this location, non-hematopoietic stroma cells provide a contact-dependent signal and growth factor required for continuous cycling of hematopoietic stem cells. The common myeloid lymphoid progenitor give rise to the sum of myeloid cells and to committed precursor of T and B cell natural killer cells lineage. The common lymphoid megakaryotic erythroid progenitor give rise to committed precursor of erythroid megakaryotic granulolytic and the monolithic lineage, which give rise to respectively to mature red blood cell, platelets, granulocyte, neutrophil, eosinophil, and basophil, and monocyte. As discussed earlier, the most dendritic cell arise from precursor that also give rise to the monocyte. The immature mast cell progenitor arise from commonly granulocyte monocyte precursor, live bone marrow, and mature in mast cell in peripheral tissue. The proliferation and maturation of precursor cell in the bone marrow are stimulated by cytokine. Many of these cytokines are called colony-stimulating factors because they were originally assayed by their ability to stimulate the growth and development of various leukocytic and erythroid colons from marrow cells. Hematopoietic cytokines are produced by stroma cell and macrophage in the bone marrow, thus providing the local environment for hematopoiesis. They are also produced by antigen-stimulating T lymphocyte and cytokine-activated and microbe-activated macrophage, providing a mechanism of increasing leukocyte production when needed for immune and inflammatory reaction and refreshing leukocytes that may be consumed during reaction. The names and properties of the major hematopoietic cytokines are listed in Table 2.7. In addition to self-renewing stem cells and their differentiation progen, the marrow contains numerous long-lived antibody-secreting plasma cells. These cells are generated in secondary lymphoid organ as consequences of stimulation of B cell by antigen and helper T cell, then migrate to the bone marrow. In addition to some lived memory T lymphocyte migrate to and may reside to the bone marrow. So hematopoietic cytokine for immune cell, there are many. So we are having cytokine, the size, the principal cellular source, the principal immature cell target, the principal cell population induced. Like the stem cell factor, CK ligands, size is 24 kilodalton, found in bone marrow stroma cell, hematopoietic stem cell, the principal immature cell target, principal cell population induced is all. Interleukin 7 has 25 kilodalton, fibrous bone marrow stroma cell, that the principal cellular source, principal immature cell, immature lymphoid progenitor. They are mostly principal cell population induced is T lymphocyte. We have interleukin 3, mostly found in T cell. The immature progenitor, they are found in all. There are many other cells that you can make follow up in the book by looking specifically to the table number 2.7. Thymus. The thymus is the site of T cell maturation. It is a bilobed organ situated in anterior mediastinum, which involves after puberty, so that is not detectable in adults. Each lobe is divided into multiple lobules by fibrous septa, and each lobule consists of an outer cortex and the inner medulla. The cortex contains a dense collection of bone marrow-derived T lymphocyte, and the lighter staining medulla is more sparsely populated within lymphocyte. The medulla also contains macrophage and dendritic cells. Scattered throughout the thymus are epithelial cells which have abundant cytoplasm. The epithelial compartment of thymus is derived from invagination of the ectoderm in development of neck and chest of embryo, forming a structure called brachial pouches. Thymic cortical epithelial cells produce interleukin 7, which is required early in T cell development, and these cells also present self-antigen to development T cell during their maturation. A different type of epithelial cell found only in medulla, called medullary thalamic epithelial cells, plays a special role in presenting self-antigen. These are medullary thalamic epithelial. So they present self-antigen to developing T cell and causing their elimination. This is one mechanism of ensuring that immune system remain tolerant to self-antigen, and it is discussed in chapter 15. In the medulla, there are structures called the Hassel's capsule, which are composed of tightly packed halls of epithelial cells that may be remnant of degenerating cell. The thymus has rich vesicular supply and afferent lymphatic vessel that drain into the mediastinal lymph node. An inherited disorder of T cell immunity caused by failure of development of thymus is called DGeorge syndrome. These patients suffer from T cell deficiency because a chromosomal deletion that eliminates gene required for thymus development. In the node mouse strain, which has been widely used in immunology research, a mutation in the gene encoding a transcription factor called TBX1 causes a failure of differentiation of certain types of epithelial cell that are required for normal development of thymus and hair follicle. Consequently, the mice lack T cell and their hairs. These mice have been used for research studies analyzing the concept of T cell deficiency. The lymphocyte in the thymus, also called thymocyte, are T cell at various stages of maturation. The most immature cell from the bone marrow enter the thymus via the blood, and their maturation begin in the cortex. As the thymocytes mature, they migrate toward the medulla. So the medulla contains mostly mature T cell. Only mature naive T cell exit thymus and enter the blood and peripheral lymphoid tissue. The details of thymocyte maturation are described in chapter 8. The thymus involutes after puberty, hence the output of mature T cell decreases progressively with age. Lymphatic system. The lymphatic system consists of specialized vessels called lymphatics that drain fluid from tissue. The lymph node is interspersed along the vessel. Lymphatics are essential for tissue fluid homeostasis and for immune responses. Interstitial fluid constantly formed in all vascularized tissue by movement of filtrate of plasma out of capillaries, and rate of local formation can increase dramatically when tissue is injured or infected. The skin, epithelial, and parenchyma organ contain numerous lymphatic capillaries that absorb this fluid from space between tissue cells. Lymphatic capillaries are behind-ended vesicular channel lined by overlapping lymphatic endothelial cell without the tight intercellular junction or continuous basement membrane that are typically of blood vessels. Lymphatic vessels are attached to extracellular matrix by elastin fiber, which serve to pull them open when there is excess fluid accumulation and tissue swelling. These vessels permit free uptake of interstitial fluid and overlapping arrangement of endothelial cells, and one-way valve with their lumen prevent backflow of the fluid. The absorbed fluid, called lymph, is pumped into convergent, progressively large lymphatic vessel by contraction of perilymphatic smooth muscle cells and pressure exerted by movement of the musculoskeletal tissue. These vessels merge into afferent lymphatic that drain into lymph node, and the lymph drain out of node through efferent lymphatic. Because lymph nodes are connected in series by lymphatic and afferent lymphatic exiting one node may serve as afferent vessel another. The efferent lymph vessels at the end of lymph node chain join other lymph vessels, eventually culminating in large lymphatic vessel called the thoracic duct. Lymph from the thoracic duct is emptied into superior vena cava, thus returning the fluid to the bloodstream or stream. Lymphatic from the right upper trunk, right arm, and right side of the head drain into right lymphatic duct, which also drain into superior vena cava. Approximately 2 liters of lymph are normally returned to circulation each day, and disruption of lymphatic system by tumor or some parasitic infection may lead to severe tissue swelling because of fluid accumulation. Lymphatic vessel collecting microbial antigen from this port of entry and deliver the antigen to lymph node where they can stimulate adaptive immune response. Microbes enter the body most often through the skin and gastrointestinal and respiratory tract. All these tissues are lined by epithelial barrier that contain dendritic cell, and all are drained by a lymphatic vessel. The dendritic cell capture the microbial antigen and enter lymphatic vessel through gaps in basement membrane. The migration of dendritic cell into lymphatic and then to specific location in lymph node is guided by chemokines produced by lymphatic endothelial cell and stroma cell in node, discussed in detail in chapter 6. Other microbes as well as soluble antigen may enter lymphatic independent of dendritic cell. Lymph node. Lymph nodes are encapsulated vascularized secondary lymphoid organ with anatomical features that favor the initiation of adaptive immune response to antigen carried from tissue by lymphatics. Lymph nodes located along the lymphatic vessels act as filter that sample the lymph for soluble and dendritic cell associated antigen. The capturing antigen can then be seen by cell of the adaptive immune system. There are approximately 500 lymph nodes in the human body. A lymph node is surrounded by fibrous capsule, beneath which is a system of sinuses lined by reticular cells, cross-bridged by fibrin of collagen and other extracellular matrix protein, and are filled with lymphocytes, macrophage, dendritic cell, and other cell types. After afferent lymphatic empty into subscapsular sinuses, the lymph may drain from there directly to the connected medullary sinuses and then out of lymph node through the efferent lymphatic. Macrophage in the subcapsular sinus provide an important function of phagocytizing and removing infectious agent, which they can be recognized by a wide variety of cell surface receptor. Beneath the inner floor of subcapsular sinuses in lymphatic-rich cortex, the outer cortex contain aggregated of cells called follicula that are populated mainly by B lymphocyte. The cortex around the follicles, called the parafollicular cortex, paracortex, or T cell zone, is organized into cord with abundant extracellular matrix protein and fiber, and is populated mainly by T lymphocyte. The development of lymph nodes, as well as other secondary lymphoid organs, depend on lymphoid tissue inducer cell and coordinated action of several cytokines, chemokines, and transcription factor. During fetal life, lymphoid tissue-induced cell, which are subset of ILC discussed earlier, stimulate development of lymph node and other secondary lymphoid organ. This function is mediated by various proteins expressed by inducer cell, the most thoroughly studied being the membrane-bound heterodimer molecule LT alpha beta 2. Mice with genetic deletion of either LT or beta do not develop lymph node or secondary lymph node tissue in gut. Splenic white pulp development is also disorganized in these mice. Lymphotoxin produced by inducer cells stimulates stroma cell in different locations of developing secondary lymphoid organ to secrete chemokines that help to organize structure of lymphoid organ. Anatomic organization of B and lymphocyte. B and T lymphocytes are sequenced in distinct regions of the cortex lymph nodes. B cells are found mainly in a follicle in the cortex. Some follicles contain central areas called germinocenter, which stay lightly within common used historical stain. Follicles without germinocenter, called primary follicle, contain mostly mature naive B cell. Follicle with germinocenter, called secondary follicles, contain activated B cell. Germinocenter develop in response to antigenic stimulation and as a site of remarkable B cell proliferation. Selection of B cell producing high affinity antibodies and generation of memory B cell and long-lived plasma cells. Each germinocenter consists of dark zone packed with proliferating B cell called centroblast and light zone containing of cell called centrocyte that have stopped proliferating and are being selected to survive and differentiate further. Germinocenter reacting during moral immune response is described in chapter 12. T lymphocytes are located mainly beneath or more central to follicles in a paracortical cord. Naive T cells enter the T zones through specialized cortical blood vessel called high endothelial venules, as described in detail in chapter 3. And T cells are densely packed around high endothelial zones. Most 70% of the cortical T cells are CD4 cell helper cell intermingled with fewer CD8 T cells. This proportion of can change dramatically during course of infection. For example, during viral infection, there may be marked increase in CD4 T cell. Dendritic cells are also concentrated in the T cell zone of the lymph nodes where they can present antigen to T cell. Anatomic segregation of B and T cell in distinct areas of the node is dependent on chemokines, chemoattractant cytokines that are secreted by specialized cells located in each area of that direct the migration of lymphocyte. These specialized cells are fibroblastic reticular cells in the T cell zone or follicular dendritic cells in the follicle zones. Follicular dendritic cells fibroblastic reticular cells are the mesenchymal derived myofibroblasts that drive formation of secondary lymphoid organ during embryonic development and contribute in the multiple ways to function of this organ. There are several subtypes of follicular fibroblastic follicular cells located in different places within secondary lymphoid organ. Most fibroblastic reticular cells in lymph node can be identified by expression of podoplantin, a glycoprotein that may facilitate adhesion of fibroblastic reticular cell. Some type of fibroblastic reticular cell that plays central roles in maintaining structural and function of lymph node include marginal reticular cell that form subcapsular sinus floor, perivesicular reticular cell that form layer around high endothelial venules, T cell zone, follicular endothelial reticular, and direct movement of T cell and dendritic cell within lymph node. B cell zone, that direct movement of B and T cell in and out of B cell follicle. fibroreticular fibroblastic reticular cell, follicular dendritic cells are derived from fibroblastic reticular cell precursor and play an essential role in maintaining follicle structure as well as B cell activation. Chemokines are a large family of 8 to 10 kilodalton cytokines that are involved in cell motility, functioning, in development, maintenance, tissue architecture, and immune and inflammatory response. Chemokine bind to receptor on lymphocyte and other cells and stimulate cell to move up concentration gradient of chemokines. We will discuss the properties of chemokines and their receptor in chapter 3. The production of particular chemokines in different areas of secondary lymphoid organ and the expression of receptor for these chemokines determine where B and T cell reside in the organ. Naive T cell express a receptor called CCR7 that bind to chemokines CCL19 and CCL21, which are produced by T cell zones, fibroreticular region. These chemokines promote naive T cell movement from blood through the wall of high endothelial venules into the T zone. The dendritic cell that are activated by microbes also expresses CCR7, and lymphatic endothelial cell expresses CCL21. This is why dendritic cells enter the node through the lymphatic, and why they migrate to the same area of the node as they do naive T cell. Naive B cells expresses low level of CCR7 and higher level of another chemokine receptor, CXR5, which recognize chemokine CXL13 produced by B cell zone, FRC, and FDC. Thus, circulating naive B cell that enter the lymph node also through high endothelial venules are attracted into the follicle. The function of chemokine in regulating where lymphocytes are located in lymphoid organs and information of these organs have been established by numerous studies in mice. For example, CXCR5 knockout mice lack B cell containing follicle in lymph node and spleen, and CCR5 knockout mice lack T zones. Anatomic segregation of B and T cell ensure that each lymphocyte population is in close contact with the appropriate antigen presenting cell, with FDC and T cell with dendritic cell. Furthermore, because of this precious segregation, B and T lymphocyte population are kept apart until it is time for them to interact in functional way, as we will see in chapter 9 and chapter 12. After stimulation by protein antigen, B and T cell change their expression of chemokine receptor and begin to migrate toward one another in response to signal from chemokine and other mediator. Activated T cell either migrate toward follicle to B cell or exit the node and enter the circulation. The activated B cell migrates into germinal center and after differentiating into plasma cell may home to bone marrow. T cell zone, FRC, maintain lymph node structure and function by forming a network of specialized tube-like structure called Frc Conduit. These conduits range in diameter from 0.2 to 3 micrometer and contain organized array of extracellular matrix molecules secreted by FRC, including parallel bundle of collagen fiber embedded in meshwork of fibrin microbes, all tightly surrounded by basement membrane produced by sleeve of FRC. The Frc conduit display chemokines on their surface and act as tracker along with T cell and dendritic cell migrate, responding to the chemokines. The Frc conduit also carries some antigen that enter the lymph node through afferent lymphatic into T cell zone for access to antigen presenting dendritic cell. The conduit begin at subcapsular sinus and extend both medullary sinus lymphatic vessel and cortical high endothelial venules. Antigen transport through lymph node. Lymph-borne substance that enter the subcapsular sinus of lymph node are sorted by molecular size and delivered to dendritic cell, macrophage, and FDC to initiate T and B cell response. The flow of subcapsular sinuses is constructed in a way that permits cells in the sinus to contact or migrate into underlying cortex but does not allow soluble molecule in the lymph node to freely pass into cortex. Microbes and highly molecular weight antigen are taken up by sinus macrophage and presented to the cortical B lymphocyte just beneath the sinus. This is the first step in antibody response to this antigen. Low molecular weight soluble antigen are transported out of sinus through Frc conduit and pass to the resident of cortical dendritic cell located adjacent to conduit. The resident of dendritic cell extend process between cell lining and conduit and into the lumen and use the process to capture ingest the soluble antigen that have entered the conduit. This path of antigen delivered may play a role in initial T immune response and some microbial antigen, but larger and sustained response require delivery of antigen to the node of tissue of dendritic cell discussed in chapter 6. Spleen is highly vascularized organ whose major function are to remove aging and red blood cells and particles such as immune complexes and opsonized microbes from circulation and initiate adaptive immune response to blood-borne antigen. Spleen weighs approximately 150 grams in adult and is located in left upper quadrant of the abdomen. The splenic parenchyma is divided into the pedipalp, which is composed of mainly blood-filled vesicular sinusoid and lymphocyte-rich white pulp. Blood enters the spleen through the single splenic artery that pierces the capsular epithelium and divides into the progressive small branches that remain surrounded by protective and supported by fibrous trabeculae. Some of the arterioles branches of the splenic artery and an extensive vesicular sinusoid that are filled with large number of erythrocytes aligned with the macrophage and other cells. Sinusoid end in venules that drain into the splenic vein, which carries blood out of the spleen into the portal circulation. The red pulp macrophages serve as important filter of the blood, removing microbes, damaged cells, and antibody-coated opsonized cells and microbes in the individual lacking spleen are susceptible to disseminated infection with the encapsulated bacteria, such as pneumococcal and meningococcal. This is likely because such organisms are normally cleared by opsonization and phagocytosis, and the spleen is an important site for antibody production. Both functions are defective in absence of spleen. The white pulp contains cells that mediate adaptive immune response to blood-borne antigen. In the white pulp are many collections of densely packed lymphocytes, which appear as white nodules against the background of the vesicular sinusoid. The white pulp is organized around central arteries, which are the branches of splenic artery distinct from branches that form the vesicular sinusoid. Several small branches of each central artery pass through a lymphatic-rich area and drain into the marginal sinus. A region of specialized cells surrounding the marginal sinuses called marginal zones form a boundary between the red pulp and white pulp. The architecture of the white pulp is analogous to the organization of lymph node, with the aggregated T and B cell zone. The central arteries are surrounded by a curve of lymphocytes, most of which T cells. Because of their anatomic location and morphological, call these T cells zone periarteriolar lymphoid tissues. B cell-rich follicles occupy the space between the marginal sinuses and periarteriolar sheath. In lymph nodes, the T cell area in the spleen contains network of conduit lined by FRCs. The marginal zone just outside the margin sinuses is distinct region populated by B cells and specialized macrophage. The B cells in the marginal zones, known as marginal zones B cell, are functionally distinct from follicular B cells and have limited repertoires of antigen specificity. The architecture of this white pulp is more complex in humans than mice, with both inner and outer marginal zones and the perifollicular zones. Antigen in the blood are derived into the marginal cells by circulating dendritic cell and are sampled by macrophage in the marginal zone. The anatomic arrangement of antigen-presenting cell, B cell together with T cell in the splenic white pulp promote interaction required by efficient development of humoral immune response, as well as we will discuss in chapter 12. The segregation of T cells in the periarterial lymph node sheaths and B cell in the follicular and marginal zone is dependent on the production of different cytokine and chemokine by stroma cell in these different areas, analogous to the case of lymph node. As in lymph node, the chemokine CXL13 and its receptor CXR5 are required for B cell migration into follicles, and CCL19 and CCL21 and their receptor CCR7 are required for naive T cell migration. To the peripheral sheath. The production of these chemokines by non-lymphoid stroma cells such as FRC is stimulated by cytokine lymphotoxin. Cutaneous and mucosa immune system. All major epithelial barrier of the body, including the skin, gastrointestinal mucosa, bronchial mucosa, have their own system of lymph nodes, non-capsulated lymphoid structure, and diffusely distributed immune cells, which works in coordinated ways to provide specialized immune response against the pathogen that enter those barriers. The skin-associated immune system has evolved to respond to the wide variety of environmental microbes. The component of this immune system associated with gastrointestinal bronchial mucosa are also called the mucosa-associated lymphoid tissue, and are involved in immune response to ingested and inhaled antigen and microbes. The skin and mouth contain large proportion of cells of innate and adaptive immune system, and important features of these epithelial tissues that they are densely populated with the commensal microbes, some of which are essential for normal physiology. The immune system in this tissue has evolved not to eliminate the commensal. We will discuss the special features of this epithelial barrier immune system in chapter 14. In summary, the anatomic organization of the cell and tissue of the immune system is of critical importance for the generation of effective innate and adaptive immune responses. This organization permits the rapid delivery of immune cells, including neutrophil and monocyte, so to site of infections, and permit small number of lymphocytes specific for any antigen to locate and respond effectively to antigen, regardless of where in the body antigen is introduced. The cell that perform the majority of effector function of innate and adaptive immunity are phagocytes, including neutrophil and macrophage, the mast cell, the basophil, the eosinophil, dendritic cell, inert lymphoid cell, inert lymphoid cell, natural killer cells, and the lymphocytes. Many surface molecules are differentially expressed on distinct types of and subsets of immune cells, and these named according to the class of differentiation nomenclature. Neutrophil, the most abundant blood leukocyte with distinct multi-lobed segmented nucleus and abundant cytoplasmic lysosomes granules, are rapidly recruited in the site of infection and tissue injury, where they perform phagocytic and microbial killing function. Tissue-resident macrophage are essential cells that detect microbial and allergy immune system and may perform other specialized function in different tissues such as lung, spleen, and liver. Monocytes are circulating phagocytes that are recruited in site of tissue infection and injury, where they are rapidly differentiated into macrophages that ingest and kill microbes, and dead host cells and secrete cytokines and chemokines that promote recruitment of leukocytes from the blood and initiate repair of damaged tissue. Dendritic cells are bone marrow derived cells with extended cytoplasmic process that are present in the tissue of the body and functioning as innate sentinel cells and the antigen presenting cell, uniquely capable of activating naive T lymphocyte. There are different subsets of dendritic cell with a different function in innate and adaptive immunity. Innate lymphoid cells are cytokine-producing cells of innate immune system with the lymphocytic-like morphology. They perform functions similar to CD4 and CD8 effector T cell. And innate lymphoid cells, which include natural killer cells, do not express highly diverse clonal-distributed antigen receptor. B and T cells, lymphocytes express highly diverse and specific antigen receptor, and they are cells responsible for specificity and memory of adaptive immune response. Both B and T lymphocytes arise from common precursor in the bone marrow. B cell develop and proceed in the bone marrow, whereas T cell precursor migrate to mature in the thymus. After maturating, B and T cell leave the bone marrow on some enter the circulation and populate the peripheral lymphoid organs. Naive B and T cells are mature lymphocytes that are not been have not been previously stimulated by antigen. When they encounter antigen, they proliferate and differentiate into effector lymphocytes that have function to protective immune response, effector B cells, and antibodies. Secreting plasma cell. Effector T cell include cytokine secreting CD4 helper T cell and CD8 cytotoxic lymphocytes. Some of the progens of antigen-activated B and T lymphocytes differentiate into memory cells that survive long period in a quiescent state. The memory cells are responsible for a rapid and enhanced response to subsequent exposure to antigen. The organ of immune system may divide into the primary or generative lymphoid organ, bone marrow, and thymus, where lymphocytes mature, and secondary or peripheral lymphoid organ, lymph nodes, spleen, and mucosa and a cutaneous immune system, whereas lymphocytes are activated by antigen. Bone marrow contain the stem cell for all blood cells, including lymphocyte, and is the site of maturation for all these cells types except T cell, which mature in thymus. Extracellular fluid, lymph node is constantly drained from tissues through lymphatic into lymph node and eventually into blood. Microbial antigen are carried in the soluble form and within the dendritic cell in the lymph to lymph nodes, where they are recognized by lymphocyte. Lymph nodes are encapsulated secondary lymphoid organ located throughout the body along lymphatic, where the naive B and T cells respond to antigen that are collected by lymph node from peripheral tissue. The spleen is an encapsulated organ in the abdominal cavity where senescent or opsonized blood cells are removed from circulation, and in which lymphocytes respond to the blood-borne antigen. Lymph node and white pulp of spleen are organized into B cell zone, follicular, and T zone. The T zones are also the site of residence of mature dendritic cell, which are antigen-presenting cell for activation of naive T cell. Fibroblastic reticular cell are specialized myofibroblastic found in secondary lymphoid organ, which are essential for structural organizational and function of these organs. Fibroblastic reticular cell produce chemokine and form a fibroblastic reticular cell conduit, both of which are required for the directed movement of lymphocyte and dendritic cell within organ, and the separation of B and T cells. Reticular dendritic cells are related to fibroblastic reticular cells and required by B cell follicle structure and function. The development of secondary lymphoid tissue depend on cytokine and lymphoid tissue inducer cell. Thank you very much. Have a look for more reading, selected reading, and you can pass through as you prepare for the next chapter, which is leukocyte circulation and migration into tissue. Thank you.